Hi everyone, thank you so much for inviting me to the IPC conference 2024. I do apologise for not being there in person. I've had work commitments that have kept me from coming to the conference, but I hope you are all having a great time. So today I'm talking about chlorine disinfection. So chlorine disinfectants versus superbugs. It's had a lot of headlines, a lot of interesting controversy around this. And it's really important for me, I feel, to put some of the queries to bed and trying to answer some of the queries that have come to me based on the research that I've done. So I'm Dr. Tina Joshi. I'm an associate professor of molecular microbiology at the University of Plymouth, and I'm an international expert in antimicrobial resistance, and I work on C. difficile spores. The presentation is C. difficile spore survival after recommended exposure to chlorine-releasing agents. Briefly, if you're talking about Clostridium difficile spores or C. diff spores, as everyone knows them, C. diff is a causative agent of C. difficile infection. And its spores are intrinsically resistant to heat, biocides, and damage. That's something sort of very, very well known. The spores themselves have an exosporium on the surface or on the outside, which you can see in this image here, the scanning electron microscopy image here. And different spores look different under the microscope, and they can adhere to surfaces very, very well. So these are very challenging to disinfect in clinical environments and, of course, on varying surfaces. Studies are showing now that C. difficile has got an increase in antimicrobial resistance, and that's important to note, definitely clinically, because it means that now we are experiencing more challenges with C. difficile itself as an organism when it infects people, but also when we're looking to disinfect C. difficile efficaciously. The AMR in C. difficile or antimicrobial resistance is really that's emerging is fidaxomicin-based resistance, and there's papers on that. Metronidazole-based resistance with a PCD metroplasmid, and some other methodologies there with metronidazole. If we want to disinfect C. difficile effectively, we have to make sure that our infection prevention and control measures are fit for purpose. We need to make sure that they are effective against the AMR C. difficile that is coming through and being more visible, definitely with patients and definitely from the laboratory. There have been increased cases and incidents of C. difficile after COVID-19. And what's interesting is that the government UK guidelines, which haven't been updated specifically since 2012, still suggest that UK guidelines should be 1,000 parts per million to 10,000 parts per million of chlorine releasing agent to disinfect any surface contacted with C. difficile for a time of 10 minutes. And the CRA or chlorine releasing agent should either be NADCC, which is sodium dichloroisocyanurate, or sodium hypochlorite. So we both know, or we all know, we both know that NADCC and NaOCl are most used to disinfect C. difficile spores. No studies have figured out the exact mechanism of action of this, though. We think that it's something to do with oxidization of the spore, but no scientific studies have actually elucidated this. We again know that microorganisms evolve, but what we don't understand is why there's a huge gap in scientific literature regarding C. difficile spore infection. There isn't much today about the mechanisms of action of some of these chlorine releasing agents. No studies that show exactly what happens in varying scenarios with these agents, and therefore I think it's very important for us scientifically to address those gaps and to address those gaps in literature. The majority of studies really focus on one microorganism in terms of spore biology, and that microorganism is Bacillus subtilis. So there are many studies from 1980 to the 2000s that focused on B sub only. And our current guidelines for disinfection of C. difficile are all based on Bacillus subtilis. So the question is, are our current disinfection guidelines fit for purpose for C. difficile? And that's a question that I wanted to answer biologically. Let's put this into context first. What's the difference between B. subtilis and C. difficile? Well, looking at C. difficile, it's anaerobic, of course. It's a causative agent of CDI, which is healthcare acquired. It's highly resistant to desiccation, as I mentioned before. We have AMR strains, and it has variations in this exosporium outer layer that you can see here. The thing is with B. subtilis, all the studies have been done on B. subtilis, but we don't really seem to understand why that's being used as a substitute for C. difficile, when B. subtilis, in terms of its biology, is completely different. It's aerobic. it's foodborne, and there's limited antimicrobial resistance or clinical impact. It's even got to the point now where we know scientifically that C. difficile and B. subtilis spores have different ways of germinating, sporulating, and developing their spores. So they have different niches and different responses to biocides. So it calls into question why we are basing all of our disinfection protocols on B. subtilis when C. difficile is a completely different beast. With that in mind, many years ago, I started studying C. difficile and found it a very interesting organism for many reasons. And one of the first studies I did in 2012 was looking at C. difficile's ability to adhere to surfaces. And this is important because what we did was test 21 strains of C. difficile, produce spores for all of them, and see if they adhered well to stainless steel, so inorganic surfaces that may be clinically relevant, and then human gut epithelial cells, which are also organic, so almost mimicking how well they adhere to a gut cell in a C. difficile infection. The spores that stuck well to stainless steel and human gut cells had a very strong exosporium layer, which you can see in figure C on the screen. It's the outside layer of the spore that seems quite crystally. That is the exosporium, and that confers stickiness. And that's important because it allows the spore to stick to any surface particularly well. And it's very hard to get rid of the spores once they're stuck on. This particular study, and you can see here the range of spores that we tested from different isolates, different ribotypes of strains as well. And of course, what you can see is variations in the ability of these spores to stick. It highlights the importance of being able to test more than one or two C. difficile strains to come to some conclusions about that organism. So this study looking at 21 strains of the organism really solidly showed us that, yeah, C. difficile strains don't all stick in the same way to surfaces. So there's variations within the organism itself. The next study we did was looking at 21 strains of C. difficile in response to NADCC, and that's the commonly used hospital biocide, as everyone in the audience probably knows, sodium dichloroisocyanurate. So you have 21 strains responded differently to sublethal concentrations of NADCC. So what I did was generate spores all at the same concentration of C. difficile from all the 21 strains, expose them to 500 ppm of NADCC. And crucially, and one thing I haven't mentioned is whenever a biocide testing is done in a laboratory scenario, we always neutralize the biocide activity after the set contact time. So that contact time, according to government guidelines in the UK, is 10 minutes, and that's usually the manufacturer's guidelines. So technically, when a biocide is administered, it should be on a surface for 10 minutes before it is wiped off. I don't know if that happens in clinical practice. There are time constraints, but that is how these biocides should be used. There should be physical removal of them from the surface of whatever surface it is. So we've got these strains, we exposed them to 500 ppm NADCC, and what we found is it changed the spore adherence ability. So the exosporium layer on the outside of the spore became more sticky with some strains after biocide exposure. And what that said to me was, okay, the biocide may not be having the effect we want it to. If you don't put the biocide on for the appropriate contact time, because we did this with 1 minute, 5 minutes, and 10 minutes, it may cause more stickiness of the spores. So you need to adhere to the right contact time. And even after 10 minutes, we may still be getting more sticky spores that aren't completely dead. So what we want to try to do with using biocides, of course, is to kill the spore. We're not seeing that in the results from 2017, and that was with sublethal concentrations. And clearly, for anyone who's following, of course, you wouldn't see that for sublethal concentrations. But what it does tell you is there are interesting variations in the spore biology between strains. So it posed another question, which is, how well do spores adhere to soft and hard surfaces? I've done some preliminary studies that have been published in 2012, 2017, as you've seen, where we've looked at spores adhering to hard surfaces like stainless steel, and then other inorganic surfaces like gut cells. But what about soft surfaces? What about things like PPE? Now, Just to clarify, there are no guidelines globally, EPA or UK based, that cover the disinfection of soft surfaces with C. difficile. So that means any bedding, mattresses, or PPE contaminated with C. difficile has no guidelines that being able to deal with it. Of course, there have been many points are made to me about autoclaving being efficacious for disinfection of C. difficile, but as you can be seen at the bottom of the page are two particular papers. Two UK studies found that C. difficile spores can survive the laundering process at 121 degrees Celsius. Now that's serious, because if autoclaving isn't efficacious and the spores are surviving desiccation, which we biologically know they can, but that's happening in a clinical environment, there's a lot of potential for transmission. So autoclaving's great, but it might not necessarily be the silver bullet that can kill C. difficile. And of course, as you'll see at the end of this presentation, there are varying algorithms that can be used to disinfect C. difficile, and I would recommend that that's where we need to go if we want to disinfect C. diff efficaciously in a clinical environment. But moving back, yes, okay, autoclaving is potentially used for soft surfaces. And we started doing more studies based on some of the other papers we read. And in 2022, we published a study to see whether single-use PPE harboured C. difficile spores. Now, I know what everyone's probably going to say. Why test single-use PPE? Surely once you've used it, you throw it away the first time. Now, we collaborated with a hospital in the US, and their personnel, their clinical personnel and healthcare workers were not throwing their single-use PPE away after they'd worn it. Be it for cost considerations, whatever the reason was, there was a lack of compliance, and that had detrimental consequences, because actually it meant that C. difficile was transmitting around the hospital. And how do we know that? Well, what we did was we got the single-use PPE from our American counterparts. We tested the PPE for the presence of spores, and we tested about whether the spores were actually present on the PPE. What we did find was that C. diff, ribotype RT027, was present on the single-use PPE, which clearly indicated that the PPE was potentially a vector of transmission if hospital staff were not complying with disinfection procedures as we were told by the IPC teams. And that's serious, because it just shows the importance of IPC compliance in and amongst the issue of disinfection. Now, moving forwards, thinking about that and the fact that these particular soft surfaces are formites, it means that we need to be very careful about how we are disinfecting these soft surfaces and how we are disposing of PPE. So again, it posed further questions, and as scientists, we want to answer these questions. So the questions next were, well, do these PPE surgical gowns contribute to C. difficile transmission? And this is a study that we published in 2019. And what we did was we deep dived into 3 clinically relevant strains of C. diff. R20291, which is known as the Stoke-Mandeville strain, RT027 ribotype, DS1748, which is a 002 ribotype, and DS1813, which is an 027 ribotype. And what you can see on your screen in these scanning electron microscopy pictures is the individual spores and just how different each one of them looks. So even under a microscope, they look different. What we did with the same hospital is do a small study with the US, and we examined the ability of these particular spores, when artificially spiked onto these gowns, to adhere to these gowns. The question was, were these gowns a reservoir for C. difficile spore transmission? In the previous study, we saw that RT027 was stuck onto these gowns. We assumed that these gowns are fomites and contributing to CDI transmission. But what happens in a laboratory scenario? So we examined the transmission of these spores between fomites, which were stainless steel and hospital floor vinyl as well. Here's what we found. We found these PPE polypropylene gowns contributed to spore transfer across from the actual gown itself to stainless steel and from the gown itself to vinyl. And what you can see on your slide right here in front of you is the graphs actually showing the presence of purified and unpurified spores of C. difficile from these strains and their ability to survive and transfer from varying surfaces. So we use a varying touch pressure to do this and to figure out whether actually these spores were moving, and the touch pressure that we use is a force of 100 grams in a specific assay that you can see within the paper itself, which was published in 2019. We managed to recover spores from each surface, and the spore types behaved differently and clearly transferred very well. We then tested the ability of these spores to survive 1,000 parts per million of NADCC, which they did, and the results are in that paper. And then we seemed to try and determine whether the spores are still viable, and they were viable. And a really nice way of showing this, apart from another graph, is looking at a scanning electron microscopy image. And what you can see before is spores of C. difficile on varying surfaces and on specific fibers of the ground. And then afterwards, you can still see the spores. And when we cultured them, they were still viable. So what is this telling you? This is telling you that C. difficile spores can adhere to varying gowns that are supposed to be hydrophobic, which means that these spores have an ability and an affinity to adhere to varying surfaces more than what we ever thought. Secondly, it's telling you that these spores can survive exposure with sodium dichloroisocyanurate, or NADCC, at 1,000 parts per million. And that's quite a high level. So now it led to a different question. And that was, well, do C. difficile spores exhibit high tolerance to chlorine-releasing agents? And it appears that they do. So what we did next was, after the 2019 study, we decided to test those same three strains on sodium hypochlorite. Now, why is this important? Well, when I released the NADCC study in 2019, I had a lot of backlash from the American media and American IPC clinicians and varying places across the globe. And the overarching remark was, well, you didn't do this with sodium hypochlorite, therefore your results aren't relevant. So I went off and did it with sodium hypochlorite. And again, these are laboratory experiments to prove a biological phenomenon. And it looks like this biological phenomenon is occurring. So what we've got here is we repeated the study using hypochlorite, used the 3 strains at 3 different biocide concentrations, 1,000, 5,000 and 10,000 parts per million. We did this in liquid, on soft surfaces like surgical scrubs, and these are the actual scrubs themselves now, not PPE that's single use, real surgical scrubs and patient gowns, and spiked the spores onto these at a range of concentrations. And this is the paper that has caused a lot of controversy, and the results that caused a lot of controversy most recently when it was published in November. So what we found was C. difficile spores do survive exposure to sodium hypochlorite in this laboratory scenario. We found that they survived exposure to 1,000, 5,000 to 10,000 parts per million, a very high concentration of spores. So we put about 1 times 10 to the 8 or 9 spores in. And we did that on purpose just to see what's happening, what kind of phenomenon was occurring. And we saw that the spores seemed to survive, and they did survive these high concentrations of hypochlorite. So this is on spike surfaces, on the gowns, in liquid, and they were viable. And we saw the electron microscopy images afterwards, which showed very limited differences in morphology of the spores. So what does that mean? It basically means that the spores are acting in a way that we don't understand. We don't know if they're creating a shield, if they're forming some kind of way of resisting the biocide, if the biocide is not working effectively because the exsporium layer is getting involved. There are many questions biologically that we don't understand. What we do know is at this concentration of spores, the biocides did not work. So that led on to other work. We also took at the same time, and this was about in 2022, the same time that we did this work ourselves, a collaborative biophysical study with colleagues at Umeå University in Sweden. And we wanted to understand the same process, why these C. difficile spores could tolerate disinfection with hypochlorite. And we did this study using Raman spectroscopy and different methodologies of creating the spores, which we think has given us some varied results. So that tells you that the way we create spores in the laboratory needs to be unified for us to make conclusions as well. Our latest paper that was just published in 2023 builds on the whole body of research that was done before, and it sought to understand how and why we had observed chlorine resistance in these C. diff spores. As yet, we haven't explored genomic resistance, although literature does suggest there are genes of interest that we can pursue that may be involved in resistance to these particular biocides. I've had queries about why we didn't use EN17126 as a test, and that's the sporicidal efficacy test for certain products, and it's an industry standard. Well, we didn't do that because we're not doing a study on sporicidal efficacy. We're trying to understand the biological process of why C. difficile is becoming resistant. These are scientific questions we're trying to understand, which will have clinical implications potentially later on. But at the moment, if we don't understand what's going on, then how can we fully translate that to clinic? So it's important for us to answer and understand these fundamental questions. Our future work involves whole genome sequencing, collaborations with hospital studies, and looking at efficacy of other disinfectants against resistant C. diff. So these include long-term exposure experiments, etc., with use of things like peracetic acid. And this is new data now. So lucky you're seeing new stuff coming from the laboratory. And really briefly, I just wanted to show you, and because of the amount of skepticism and pushback I've received from the C. diff and varying communities about this resistance or tolerance to biocides, we've done longer experiments, a whole range of generational passages and exposures of these spores to varying concentrations of biocides. And what we've seen moving forward is, again, survival of C. difficile across varying passages with 2 NADCC at varying concentrations of biocide, as you can see on the screen now. And then again to hypochlorite, which you can see on the screen now. So this isn't a unique phenomenon. We know that something strange is happening. And I think what I'd like everyone to take away from this is that C. difficile spores biologically behave differently. We can't treat them all the same way. We know that chlorine-releasing agents can change spore adherence ability to hard and soft surfaces. Spores very clearly can resist exposure to NADCC and NaOCl, so using the right biocide at the right time at the right concentration is really important to ensure efficacy of disinfection. Laboratory studies like what we've done that are observational are usually indicative of what can happen in clinical environments in the future. And just because there are no guidelines around soft disinfection of surfaces or guidelines around laboratory studies doesn't mean that it has no relevance or bearing clinically. So it's really important for us to look at guidelines for soft surfaces and disinfection of soft surfaces, check that our current guidelines for disinfection of C. difficile are fit for purpose, and that they cover the new AMR and biocide tolerant strains of C. difficile that may be coming into clinic. And of course that the EN17126 ISO test may not be fit for purpose if it only tests one strain of C. difficile that is not AMR or clinically relevant. We do need to do more studies with a range of strains, and I have just written a response to comments made of the article from Ahmed and Joshi we published in 2023. I've got a new response to comments paper coming out soon in the Journal of Medical Microbiology, which covers all of this and all the things I've talked about today. Prevention is key. You want to disinfect effectively, and ignoring scientific studies perhaps doesn't play into that particularly well. So I think it's really important for us to look at disinfection algorithms, keep our minds open, and look at disinfection alternatives like UV, hydrogen peroxide vapor, hypochlorous acids, antimicrobial surfaces, and to generally keep up to date with scientific literature, because it does bear relevance to clinical outcomes in the end. And to say it doesn't is perhaps closing the door when some of these scientific studies do have relevance and are useful. I want to thank you all for listening. Some acknowledgements to my students who are part of my research group. Thank you very much to them who've done some of the studies with me, my collaborators in the US and in Sweden, and again to the IPC committee for inviting me today to give this presentation. If you have any questions, please feel free to contact me at my email address. And again, thank you very much for your time.